fellowship, and then students can apply for funding to do research during the summer. So if you think you want to do research, apply. The applications come out around February, and then they go to a committee to evaluate the proposals, and then we'll let you know in March, and then you can start your research. Okay. So Kyra Tan, and I'm really excited to hear what she's going to say.
Hi everyone. Sorry. Hi everyone. Thanks so much for coming out today. I'm Kyra, and this summer I investigated the effects of blue and red lights on culturing phytoplankton, and I also experimented with different methods for isolating wild phytoplankton. I'd like to thank Ms. Kelly for being my mentor, and I'd also like to thank the K Fellowship for making this project possible. So when I was a kid, I loved to go to the aquarium. I used to go a lot. And I'm lucky now that even though I'm older, I still have an excuse to go every other month because my brother loves to go. He's 7. So even though his favorite thing in the world is Transformers, I think every 7-year-old can appreciate a giant 8-foot shark like swimming right past their face. But for me, my favorite animal has always been the seahorses at the aquarium. And when I was 7, I used to beg my parents for my very own seahorse pet. And they used to tell me that it was impossible because they're way too hard to care for. And I thought that was just an excuse, but they were actually right whether they knew it or not. Seahorses, part of the reason seahorses are hard to keep in captivity is because they have a very, not very developed digestive tract, so they have to eat a lot to absorb any nutrients. This can be especially true for baby seahorses and challenging because they need very tiny food. This past year, the Waikiki Aquarium actually had an exhibit on their baby seahorses, and if you looked at them, you could see just how tiny their mouths were. So this meant that the aquarium had to recreate their wild diet of like tiny rotifers, copepods, and artemia. And they started at the very bottom of the food chain, which meant culturing phytoplankton to feed the zooplankton that would feed the baby seahorses along with lots of other fish fry. So just a little bit about phytoplankton. Plankton are defined as like small and sometimes microscopic living organisms, and they're suspended in the ocean's water, and they're at the mercy of the tides. There are 2 major groups of plankton. One is zooplankton, the animal plankton that includes like small crustaceans and invertebrates and fish eggs and things like that, and then the other being phytoplankton or plant plankton. So this can be like marine algae or like diatoms, which are single-celled plant algae, like the 2 species that I worked with, Chaetoceros and Tetraselmis. So being able to recreate a fish's wild diet is really important for being able to successfully breed and raise them in captivity. Part of the reason we want to be able to do this is because it's important to the scientists who study these fish, and it's also important to wild fish populations. In the pet trade every year, on average, about 27 million fish are removed from reefs annually. Compared to about like 90% of freshwater fish species, only 10% of marine fish are able to be raised in captivity. So this makes it really, so making it possible to raise fish in captivity would really help to relieve the pressure off of wild fish populations. So my research was focused around seeing if there was any way we could help the Waikiki Aquarium provide more accurate diets for their fish. The things I set out to do were to see if phytoplankton culturing could be further optimized and to try to isolate native Hawaiian species of phytoplankton that we presume most Hawaiian fish would rely on. So for my first task, I decided to test if Tetraselmis and Chaetoceros, these 2 genuses of phytoplankton the aquarium uses a lot, could be grown better underneath red and blue lights compared to the white lights that the aquarium uses. So the reason I chose to test red and blue lights is because of the color of Tetraselmis and Chaetoceros. Tetraselmis, for example, is green, and to us, it looks green because it's reflecting green wavelengths of light the most, which inversely means that it's absorbing other wavelengths of light more, particularly like red wavelengths because it's the opposite color. So using this logic, I thought that also because Chaetoceros is like an orange brownish colored phytoplankton, it might absorb blue wavelengths of light best. So I tried to imitate the aquarium setup as closely as possible. The upper left photo is a photo of the aquarium's plankton room, and below is like my bare setup, kind of like just the air tubes and the air lines, and then the air filters and flasks. And then the two variables that I kept constant were airflow, which I used an airflow meter to control, and then nutrients, which I used F/2 or like a marine fertilizer for. So this is just a visual of my overall experimental design. I ran and collected data from a total of 18 flasks or cultures, 9 for Tetraselmis and 9 for Chaetoceros. For each trial group, I had 3 replicates, meaning I ran 3 flasks on white light, 3 flasks on red light, and 3 on blue light for both Tetraselmis and Chaetoceros. Sure. Sorry. Test? Okay. Sorry. Okay, so I collected data on their density or their cells per milliliter every other day, and I gave each flask roughly a 2-week growth period. So for cell counting, I'd use a hemocytometer, which is basically a slide with grid lines on it and a counting field, and I would count the number of cells in a sample from each flask. And I'd use like a clicker to keep track, sorry. A funny thing I wish I could show you guys is Tetraselmis, because they're actually flagellated. They have 4 little tails, and they can zoom around really fast. So in order to be able to count them, I would have to, unfortunately, stun them with a little bit of bleach to keep them from moving. So this graph is plotting the percentage change of cell density between sampling days. So my results suggested that red lights were statistically significantly better for Tetraselmis growth compared to it on white light. And blue light was not statistically significantly different, but both red and blue light had higher means of growth rate than the white light did. And then for Chaetoceros, my results suggested that red light was also significantly better for their growth rates, and that blue light was not significantly different. But both red and blue lights also had a higher mean of growth rates. So my data doesn't support my initial prediction that Chaetoceros would grow best on blue light because of its orange color, and it's possible that Chaetoceros simply absorbs red light better than blue light, or maybe it had something to do with other possible sources of air in my experiment. So some of these sources of air would include starting density. Some of the flasks didn't start with the same density of cells, so this variation might have influenced growth rates, because if individual cells had more space and nutrients to themselves, presumably they might grow and propagate more. There's also variations in light intensity. The lights that I was able to use had different light intensities, like the white light I had measured at like 53 lux, and the blue light I had measured at around 20 lux, and the red light 23. And then another variable was temperature. So for plankton, the optimal temperature, or phytoplankton at least, the optimal temperature is around 75 degrees Fahrenheit. But during the day, the AC would run and the lab would be around 75 degrees Fahrenheit. But at night, when the AC was turned off, it could rise to about like 80 to 85 degrees Fahrenheit. And then another bump I encountered was the occasional like clogged airline with salt. So if I didn't notice it in time, it might have affected how much air each culture got, and I would have to clear it. And then another error was that I didn't run all of the trials simultaneously, because I could only run so much at the same time because of space and like time limitations. And then some of the cultures crashed because phytoplankton or algae cultures can be easily infected by other algae and organisms. So I tried to avoid this by sterilizing a lot, like using alcohol to wipe down the area and acid washing all of the equipment and autoclaving. But despite this, I still had crashes and I had to rerun some of the trials. For the second part of my project, I was actually doing it at the same time, but I was looking at if I could isolate wild species of plankton. So before I could attempt isolation, the first step was to collect wild samples of plankton. And I did this by just going down to the Ala Wai and using a 20-micron plankton net and sweeping it along in the water and filtering out plankton. And there's definitely lots of plankton, both zoo and phyto. I did see Chaetoceros, and they weren't the most abundant species, but they're definitely there. As for Tetraselmis, I'm not entirely sure if I saw any because they tend to move around very quickly. And so the first method I tried was manual isolation, and it's basically you use like a tiny little glass rod that's been heated up and stretched to a fine point, and then you use that by looking underneath a microscope and trying to pull out an individual plankton through capillary action. It was a little difficult because the glass tips were really fragile, and when you're working through a microscope, all of your motions are in reverse, so recoordinating my hand was a little challenging. Unfortunately, I don't think I was successful in drawing out any plankton, but the plan was to, after I isolated a plankton, to put it into a culturing medium and letting it grow and propagate to get hopefully a homogeneous sample of that plankton. The second method I tried was agar plating. So I tried a couple of different agar recipes, like ratios of agar to the nutrients medium. And for example, like the first recipe I tried turned out a little bit too soft, and it was too fragile to properly plate the plankton. So after I figured out how to get the consistency right, I tried both using like different tools, like a bacterial loop and a bent rod to distribute the actual plankton on the plate. And I plated some controls using the plankton from the flasks I had running, the Chaetoceros and Tetraselmis. So what I was aiming for was to have a single colonies of the microalgae grow on the surface of the agar, and these colonies would hopefully be made up of all like a single species. And then I would be able to pick off the colony and then isolate it in culture medium. But I let them grow underneath lights for a couple of weeks, but unfortunately I didn't observe any growth even in my controls. There you can see my Chaetoceros and Tetraselmis positive controls, which unfortunately didn't grow anything. So there's probably something wrong with the recipes that I used, or it just didn't work for the phytoplankton that I was trying to culture. And then the last method I tried for isolation was dilution, and basically it means like taking a sample of plankton, and then you take a cell count on it, and then you calculate out like how much water and nutrients you would have to add to the solution to statistically get a single cell isolated into each test tube. So I did about maybe like 120 to 160 different test tubes, and I left it in like kind of low light conditions so that the cells wouldn't get too shocked, hopefully. Unfortunately, I haven't observed any growth yet. Microalgae, or at least marine microalgae, can take up to several months to grow from this kind of method, I've read. So I still have them, so fingers crossed, but not expecting too much. And so for my reflections, I learned a lot about lab equipment and procedures, and I got to use a sterile hood and learn how to use an autoclave machine, and also how to do things like acid washing and a whole bunch of other important like lab skills that help to reduce like injury and risk. And I also learned a lot about culturing phytoplankton. So in the future, if I ever am successful in isolating a wild species, hopefully I'll be able to actually culture it. And then, but more than that, this experience of research has been really valuable, like learning how to plan ahead, though sometimes that's a lesson learned too late, and troubleshoot and accommodate for unexpected blips that might occur. I'm really glad I had this opportunity in high school instead of college. And one of the things I learned about research is that it's often cyclic in nature, and of course you should plan ahead, but going back to the drawing board can also be a really big part of the process that's not necessarily bad. Not to mention, this summer was incredibly fun for me. I had so much fun collecting phytoplankton, growing phytoplankton, and talking about phytoplankton to all my friends. I don't know if they enjoyed that, but so my advice for those who might be interested in this project is to go for it. It's an amazing experience. And I did this while doing 3 other summer school classes, and I don't know if that's recommended. It can be a little hard, but ideally you'll have like a good understanding of the general topic and your plan for research before the summer begins. And hopefully you can try to plan for a little extra time to redesign or deal with or troubleshoot any things that might not go according to your plan. Yeah. And I think this experience, like no matter what field I go into, will always be relevant in terms of like learning how to create a project and then execute and then reflect back on it. So I'd like to share my thanks to Mrs. Kelly over here, my mentor, and Mrs. Lentes for sharing this project with me and also being such a huge help in answering all of my questions about phytoplankton. And I'd like to thank Dr. Chan for her endless patience, showing me how to use the lab and be safe in the lab, helping me plan and troubleshoot and analyze. Thank you so much. And I also have to thank my parents, who were supposed to be done carting me around in July, but I chickened out and I didn't take the road test, so now I still have my permit. And I'd like to thank Brennan He graduated last year. He had done a project on phytoplankton, or not phytoplankton, just plankton in general in the Ala Wai too. And instead of enjoying his summer, he answered a bunch of my stupid questions about plankton. And of course, special thanks to Violet and John Kay for providing me with this amazing opportunity. I've had so much fun and I've learned so much. Thank you so much. I'm happy to take any questions, or if you have questions about my project or the K Fellowship in general, feel free to email me.
Yeah, so if you have a question, just jot it down, and we're going to have Yufei go, and then at the end, we'll go through the question.
Oh wait, this is your iPad, sorry. Okay. Hi, my name is. Oh. Test. Okay. Hi, my name is Yufei, and I did my summer research on the physical oceanography of the Ala Wai. So, unlike my friend Kyra here, I focus more on the non-living aspects than the living ones. So I've always been interested in how various structures and the contents within it influence each other. Like microscopic particles and macroscopic, like wormholes and stuff, all have connections, all connect with each other through various kinds of structures. So I would really like to investigate the structure of the Ala Wai. Whoa, okay, yeah, that's good. Okay, so with that in mind, I would really like to investigate the structure of the Ala Wai in terms of how the things within it interact with each other and create such a complex system. So the Ala Wai Canal was first drained from wetlands in the 1920s, and since then it has been a very diverse bio-ecosystem, and it contains a lot of different nutrients and bacteria, and that includes both bad and good things. So the bad part, which is the pollutants and the pathogens, is a problem in our neighborhood because, well, first of all, pollutants that flow from large storms into the streams and into the canal would overflow into the ocean, and that would cause turbidity plumes in the ocean, which is not good. And also, pathogens such as Vibrio vulnificus also grow in the canal. So you can see here, the Vibrio infection rate in Hawaii is significantly higher than other states, mainly because of the growth in the canal. So knowing that the problems in the canal is very significant, we might investigate the causes to such biotic problems. So the hypothesized relationship, which has been confirmed several times, is that the climate around the canal would be the major influence on how the pollutant and pathogen levels fluctuate. So in here you can see that environmental and climate factors such as bathymetry, which is basically the shape of the land, and tides and runoffs would change the variables within the canal, which is the flow or velocity, temperature, and salinity. And these factors, with our hypothesis, would impact the pollutant and pathogen levels in the canal. So one example of this relationship would be on the right. So this is a graph of Vibrio vulnificus abundance at different salinity and temperature ranges.
So you can see that in the temperature range between 25 and 30 Celsius and the salinity between 1 to 15 parts per thousand is the best condition for Vibrio to grow, which is basically the condition in the canal. So knowing that it's really important to investigate the physical structure of Ala Wai to understand why the problems are occurring, it's important for us to look more into the structure itself. So previous studies has been done in the past on basically what the physical oceanography of the canal is. So Dr. Frank Gonzalez in 1971 has done a very in-detailed oceanographical analysis of how temperature, salinity, and flow vary in the canal in terms of patterns and trends. And that has been the basis of most of the studies done on the Ala Wai ever since, such as Ms. Emma Ness's research on the microbial models in 2015. So I thought it was important to update Dr. Gonzalez's study because a lot has changed since then due to climates and such. So my purpose in this research is to investigate the changes to the physical structure of the Ala Wai from 1971 to now. And on the side, I would also like to experience research and develop skills. So to put the purpose more into detailed slices, I split it into two questions. So one, how has structure changed from 1971 to now? And that includes temperature, salinity, and flow. And secondly, if the structure is intact, then how has the climate input and the affect the nutrient and bacteria output? So you can see here, this is a diagram I drew of the basic structure. And the inputs would include, as I said before, air temperature, wind, runoff, tides, and bathymetry. So these things would interact with the temperature, salinity, and flow structures in the canal and create different nutrient and bacterial levels. So as an example, you can look toward the right and see the big cleavage in the canal. That's the basin section. So, and you can also see that on the right, there's a very tall tip, which is the sill. So the sill, as bathymetry, would obstruct tides and flow from the streams into the basin, and therefore there's a lot of sediments and bacteria just settling in the basin because they're not being flushed out. So that's just a relationship shown. To answer these questions, I assembled several data sets. So first set is the one I obtained from Dr. Brian Powell from Physical Oceanography. And this data set contains sensor data of temperature, salinity, and velocity through the 2.5 month period. And it's recorded at very short intervals at about half a minute per data point. So it's very useful for time series data. And I use it mainly for the structural analysis. And secondly, I had the US Geological Survey data on the stream gauges, which would supply me with data on the forcings and impact of streams into the canal. And thirdly, I have CPy ties information, which is a Python programming language package that I will describe later. And also the ICAP climate briefing for general information about the climate changes. So in order to make sense of the data, I learned Python language, which is a programming language used mainly for scientists. So since I haven't had many experiences with coding languages in the past, I learned it mostly through a lot of tutorials and also communities such as Stack Overflow and GitHub. I used interfaces in Terminal and I applied the Anaconda virtual environment, and I used the IPython interface to make it easier because it color codes things and that's easier. And As for coding packages, I use numerical analysis, NumPy, oceanography statistical analysis, CPy, and matplotlib for visualization. So using Python and datasets, I transform the raw data into graphs. So this is basically a run through of one of my graphs. First, I input the packages into the interface. Then I open files. And in this particular process, I was trying to isolate the effect of tides on velocity. So I extracted the tidal information with CPy at this specific time range, and then I just graphed it, and that's what I got. And it looks really pretty. So with these process graphs, I try to answer my questions about structural change and input-output change. So I would split my summary section into like 2 parts for every subject. So it would be first structure, which would be like the things that I found that has changed in the general structure, and secondly would be what it implies about the climate onto the biotic factors. So first of all, bathymetry has changed only slightly due to dredging. So you can see from Gonzalez's bathymetry plot to now, there hasn't been very significant changes as to the sections. So you can see there's a small, tiny block here, and then you'll see the channel and the basin running to the library end. So this would mean that not much has changed to the flow structure. However, like little changes would also be interesting to look at, such as the taller sill section. You can see between the second and the third red line. So that would, as I said before, obstruct flow. So yeah, the increasing taller sill would obstruct flow, and therefore sedimentation would occur and stuff just stays down there and not move much. You can see in the middle, that's a very interesting graph made by Ms. Nuss in 2015. It was looking so strange because they hadn't had enough data on the bathymetry, so she had to estimate the structure, and she said later that it might have been an important factor to how her microbial levels came out. So yeah, it proves that it's kind of important. So the second aspect I would like to look at is temperature. So again, the structure hasn't changed much. You can see from Gonzalez's temperature trend that mostly the temperature on the ocean side would be gradually increasing towards the basin side, and that's the same case in my data. So you can see on the lower right graph that the blue line, which is the sensor placed in the basin, generally has higher temperature than the one on the seaside, which is the aqua colored one. So having this similar temperature structure, we could infer about what the input, which is climate, on the output. So from ICAP climate breathing data, whoa, okay, I'll speed up. We can see that air temperature in the past 30 years has increased for about 48.48 Fahrenheit, and that would really create a better condition for the Vibrio vulnificus to grow in, because they like warm places. So the final structure that I would like to look at is velocity. So since I didn't have similar velocity investigations as Gonzalez, I would just look at the correlation between tides and runoff on velocity. So first of all, you can see that on the raw velocity graph with the tides included, there's a very significant pattern to it, which is basically the high tides and the low tides. After you detide the velocity range would be significantly narrower, which would mean like generally velocity is slower. And second, we can look at runoffs, which does not have as much influence, but still. So the first graph is of a day in which there was a very high runoff recorded by the USGS, and the second one was with low runoff. So the one with high runoffs had somewhat more extreme peaks in velocity, which would mean that it was flowing faster. So if these two tide and runoff relationships does exist, then we can conclude with the ICAP information again that says, oh, rainfall and stream has decreased in past 3 decades. Then once tides decrease and runoffs decrease, the velocity would also decrease. And when velocity decreases, the flow in all areas would be slower, and therefore the sediments and bacteria wouldn't flush as quickly. So from the 3 structures and implications that we looked at, we would see that the general bathymetry, temperature, and salinity flow structure hasn't changed too much. So we plug that into the climate change setting that we have here, and we would see that the biotic levels would change. So again, we trace back the 2 paths. If we have lower runoff, then we have slower residence, and we will have more sediments. And this is actually confirmed by another student research done by Sarissa and Kayla, was it? Yeah, you can go listen to their symposium as well, but they proved that basin side has more organic sludges than the channel side, which means that there's more pollutant sediment just collecting there. And on the other route, we can see that if temperature rises, then the Vibrio volnificus abundance would also increase, and this is confirmed in Dr. Stewart's and Dr. Nigro's research done in UH. So in reflection to my summer, I would say that it has definitely been an experience, because I guess this is just how independence feels like. You have both the happiness and painfulness in it. So happiness would be just having been able to learn about all the different things and make some sorts of breakthroughs in my own knowledge. And the painfulness would be just the uncertainty about what I'm doing exactly, and also about various failures that's basically inevitable in this process. So, well, in looking forward, I would say that more research can be done in physical oceanography, because right now, UH physical oceanography they are using those sensor data to update the ROMS model, which is a regional ocean modeling system. So if we have a better and more accurate model, then we would be able to predict bacterial and pollutant abundance in a more accurate way, and that would hopefully help solve the problems. And for me and for other students considering doing research, I would say that, I would say that just do it and ask questions and try whatever you think, even though you might doubt it. Yeah. And I would like to thank Dr. Chan for guidance, Dr. Powell for just teaching me a lot of things, and the John and Violet K Fellowship for just providing this opportunity that I otherwise wouldn't have done so early. And everyone for staying with me thus far. Okay, thank you.
It's true. It's true.